So what is the definition of apoptosis? It is a energy dependent, it is a ATP dependent program cell death, and it involves only the single cell or the small number of cells. You can say small number of a small group of cells. So this is the definition. Why I am saying it is a single cell, it are small group of cell, because you have to compare it with necrosis. Necrosis is also the cell death program, but keep in mind the necrosis involve multiple cells. Necrosis involve large group of cells. Necrosis is not ATP dependent. Necrosis is a passive process. Apoptosis is an active process. Active process means it is dependent on ATP. Then after that, we have to keep in mind they can also ask from you the different examples. Like, can you give the different examples of apoptosis? So during the menstrual cycle, endometrium is going to shed. This is an example of apoptosis. It is a normal physiological process, because apoptosis can be physiological, can be pathological. And the physiological example is menstrual blood loss, and the second example is during the embryogenesis, there is some kind of cells which are removed from our body. Removal of cell during the embryogenesis is also the example of apoptosis. Then very very important, there is a one process in our body when the hepatocytes are get infected. There are CD8 cells in our body. These CD8 cells are going to kill the viral infected cells in the body. So this is also the process of apoptosis. But you have to keep in mind that apoptosis can be physiological but can be pathological also. So these are the examples of apoptosis. Then after that, we have to understand the apoptosis is basically, you can say, in this the cell death can occur through different mechanisms, like intrinsic pathway, extrinsic pathway. We have the porphyrin gray enzyme pathways. These are the different pathways through which the cell is going to be dead, through which the program cell death occur. So intrinsic pathway, extrinsic pathway, porphyrin or gray enzyme pathways, all these pathways are going to activate the caspases. So what are caspases? Basically, these are the enzymes, these are the proteases which are present in the cytosol. So whenever you are going to make a program for the cell death, the intrinsic pathway, extrinsic pathway, or porphyrin or gray enzyme pathway, all these pathways have to activate the caspases. If you will not activate the caspases, the cell breakdown will not occur, the cell shrinkage will not occur, chromatin will not condense, and the membrane blebbing will not occur. So that's why it's very important for these pathways to activate the caspases. And when the caspases are activated, basically these are the proteases enzyme. These are the enzymes which will destroy the our cell, which will destroy the proteins. So when these enzymes are activated, the cell breakdown will occur, and when the cell breakdown will occur, the cell will shrink, chromatin will become condensed, and the membrane blebbing will occur. So these are the you can say morphological changes which will occur in apoptosis. Then after that, they can ask from you in exam, like how to characterize the apoptosis. How to characterize the apoptosis. So apoptosis is characterized by basically deeply eosinophilic cytoplasm. So it's mean the cytoplasm of the cell is going to be red or pink. Eosinophilic mean it is going to be red or pink, and the cytoplasm will become red or pink, but the nucleus will become the basophilic blue, and the nucleus is going to be degraded with the help of pycnosis and karyoexis, which we already discussed in the necrosis. So let's try to repeat one time again. First of all, what is apoptosis? It is a ATP dependent program cell death, and this cell death involves the single cell and small group of cell. Then what are the example of apoptosis? Endometrial shedding during the menstrual cycle, removal of cell during the embryogenesis, or CD8 cell mediated killing of virally infected cells in the liver. So all these are the example of apoptosis. Then after that, you have to keep in mind what are the morphological changes. What are the morphological changes in apoptosis? So first of all, you have to keep in mind, before studying the morphological changes, we have to activate the caspases. When the caspases are activated, they will activate the proteases. When the proteases are activated, they will break down the cell will shrink, the chromatin become condensed and the membrane blabbing will occur. So after all these changes, when the membrane blabbing occur, chromatin become condensed, cell will become shrink. So this is called a formation of apoptotic body. So let's suppose this is the cell. This is the normal cell which is going to be, you can say, die with the help of apoptosis. So when the apoptosis will occur, first of all, this cell will become shrink. Its size will decrease. After that, its chromatin condense, and after that, the membrane blabbing will occur like that. Membrane blabbing will occur. So basically, this is the apoptotic body, which is going to be removed from our body with the help of phagocyte. Phagocyte. So phagocyte is going to eat this apoptotic body. This apoptotic body is only formed when the cell will shrink, when the chromatin become condensed, when the membrane blubbing occur. So these are the, you can say, morphological changes which you have to keep in mind. After that, as I already mentioned, first of all, you have to know the difference between eosinophilic and basophilic. Eosinophilic mean more pink or more red in color. Basophilic mean blue. So first of all, when the apoptosis occur, it is characterized best by the cytoplasm will become red or pink, nucleus will become blue, and the nuclear degradation will occur with the help of pycnosis and karyoexesis. So please keep these things in mind. Then after that, very, very important, cell membrane typically remain intact without significant inflammation. As I already mentioned previously, the necrosis, in the necrosis, plasma membrane will rupture. But in case of apoptosis, the plasma membrane will not rupture. When the plasma membrane will not rupture, the cytosolic enzymes will not leak. When the cytosolic enzyme will not leak, the surrounding inflammation will not occur. Okay, so here the important thing they can ask from you in the exam, what is the indicator of apoptosis? So this is very, very important MCQs they can ask from you in the exam. What is the most sensitive indicator of apoptosis? So basically, DNA leathering. DNA leathering mean basically these are the fragments of DNA which are in the multiply of 180 BP. So you guys just have to memorize this. If they ask in exam, what is the best indicator of apoptosis? So it is the DNA lathering. So just keep in mind. Then after that, we also have to complete this. Let's complete this topic. First of all, you guys have to keep in mind apoptosis can occur through three pathways. First pathway is intrinsic, second is extrinsic, and third one is the perforin gray enzyme B pathway. So first of all, what is the intrinsic pathway? We will study this. After that, we will study the extrinsic pathway and then perforin gray enzyme pathway. So first of all, intrinsic pathway is also called mitochondrial pathway. In this pathway, the tissue remodeling is involved in the embryogenesis. So just always keep this in mind. Intrinsic pathway is involved in tissue remodeling during the embryogenesis. When the embryogenesis is occurring, the tissue remodeling is going through this intrinsic pathway. And you have to keep this in mind. It occurs when the regulating factor is withdrawn from a proliferating cell population. It's mean when you are let's suppose in your body, if a factor is going to proliferate the cell, but if you remove this factor or remove this signal, then the proliferation of cell is not going to be happen. So they say it occurs when the regulating factor is withdrawn from a proliferating cell production. They give the example of decrease interleukin 2 after a completed immunological reaction, and after that it will lead to apoptosis of proliferating effector cell. So just keep in mind, when you are removing a regulating factor from a proliferating cell, after removing a regulating factor, that proliferating cell is not going to further proliferate. It will go under apoptosis. And after that, you guys have to keep in mind there are certain types of exposure factors which can also activate the intrinsic pathway. That is like the radiation, toxins, and hypoxia. So these are all the factors which can cause the apoptosis through intrinsic mitochondrial pathway. Then after that, we have to keep in mind the intrinsic pathway is regulated by BC2, BCL2 family of protein. Then just keep in mind there are two kind of proteins. One protein is going to induce the apoptosis, and second protein is going to against the apoptosis. So the protein which is going to stimulate the apoptosis, it is called pro-apoptotic, and that is Bax and Back gene. and the thing which is going to against the apoptosis, that is called anti-apoptotic, and BCL2 and the BCL-XL are anti-apoptotic. And just keep in mind the BAX and the BAX form the BAX proteins, basically these are the pro-apoptotic, and these are present in the mitochondrial membrane, and they help to release of cytochrome C from inner mitochondrial membrane into the cytoplasm, which will activate the caspases. So just keep in mind, BAX and the BAX form pores in the mitochondrial membrane, they help to release of cytochrome C from the inner mitochondrial membrane into the cytoplasm. When the when it go when the cytochrome is going from inside to the cytoplasm, there it will activate the caspases, and when the caspases are activated, they will finally destroy the cell. So just keep this in mind. Then after that, BCL2 keep the mitochondrial membrane impermeable, therefore by preventing the cytochrome C release. So it's mean BCL2 is anti-apoptotic. BCL2 is how anti-apoptotic? Because it will not allow the cytochrome C to release. And if the cytochrome C is not going to release, it is not going to activate the caspases. And if caspases are not activating, your cell death will not occur. That is very, very important MCQs. They can ask from you in exam. Give an example of any cancer in which BCL2 overexpression occur, and that is the follicular lymphoma. In follicular lymphoma, there is overexpression of BCL2, and if the cancer is showing BCL2 overexpression, it's mean the cancer cells are not going to be dead. And in this cancer, there is a translocation between the chromosome 14 and 18. So these are the chromosomal translocation which can ask from you in the exam. Then after that, if the BCL2 is overexpressed, so just try to make this concept very clear. When the BCL2 is overexpressed, your cytochrome is not going to release. When your cytochrome is not going to release, the caspases are not activated. When the caspases are not activated, the tumor will grow. The tumor will grow. It is called tumor genesis. So this is a very easy explanation. So just let me try to explain with the help of an example. So this is the mitochondria, and in the mitochondria, this is the inner membrane. In the inner membrane of mitochondria, basically there are certain kind of proteins like BCL2. Just keep this in mind. And as I already told, this is the inner mitochondrial membrane. Here the cytochrome C. If the cytochrome C will come outside from this inner membrane to this cytoplasm, here the caspases are present. When the cytochrome C will come from inner to outer membrane, it will activate the caspases, and the caspases will lead to apoptosis. So this is how they explain these things in this paragraph. After that, we have the extrinsic pathway, which is also called death receptor pathway. And this pathway is activated with the help of ligand receptor interaction. It's mean the Fas ligand bind to the Fas95 or tumor necrotic factor binding to its receptor. It's mean it is a pathway which is activated through interaction of different kind of receptor. Like when the Fas ligand bind with Fas CD95 receptor, the extrinsic pathway will activate it. And when the tumor necrotic factor alpha is going to bind to its receptor, it will also activate the extrinsic pathway. Then they say the Fas ligand interaction is necessary in the thymic medullary negative selection, and this is the important MCQs they can ask from you. Like Fas ligand interaction is necessary in thymic which selection? It is the medullary negative selection. Please keep this thing in mind. Then after that, let's suppose if in a person this Fas ligand interaction become abnormal. When this interaction become abnormal, there is the failure of clonal deletion. There is a failure of deletion of cell, and if there is a deletion of cell, those cells will self react against our lymphocytes, and when the lymphocyte will react against our own cell, they are destroying the normal person human body, you can say cells. So this is the condition which is called autoimmune lymphoproliferative syndrome. So autoimmune lymphoproliferative syndrome is what? Basically, it is a condition in which there is a defect in Fas ligand interaction. When this interaction will not occur, there is a failure of clonal deletion. Then the deletion will fail. Then there are large number of lymphocytes which will react against our normal cell. When the lymphocyte will react against our normal cell, it will lead to lymphadenopathy, hepatosplenomegaly, and autoimmune cytopenia. So these are the conditions which will develop after the lymphocyte will react against our normal cell. So just keep in mind, autoimmune lymphoproliferative syndrome will occur when the effect in the Fas and Fas ligand interaction. So this is an important MCQs. Then after that we have the porphyrin, a gray enzyme pathway. So basically this is very interesting pathway. You guys can remember, like this is the cell. Let's suppose this is the cell, and in this pathway basically the porphyrin and gray enzyme B. Let's suppose this is the porphyrin and gray enzyme B. So first of all, this is the porphyrin. This porphyrin will come and bind with the cell, and this porphyrin will try to produce the pores in the cell. Because it is called porphyrin, it will make pores in the cell. So when it will make pore in the cell, through those pores the gray enzyme will enter in the cell, and when the gray enzyme will enter in the cell, it will lead to the cell death. So they say a release of granules containing porphyrin and gray enzyme B by immune cell, and the porphyrin form a pore for gray enzyme B to enter the target cell. So it's mean simply please keep this in mind. The cell which is going to which is going to under apoptosis with the help of gray enzyme B pathway. First of all, there is a porphyrin. Porphyrin is like, it is a machine. It is like a drill machine which is going to make a hole in the cell. Through those holes, the gray enzymes will enter and lead to the cell death. So that's all about the apoptosis. So please